How's it going, everybody? Good? Hey, excellent. Welcome. Oops. Where's my clicker? Welcome to Microbes in the Modern World, from the globe to the gut. The second talk in our series this term. How many people came to the first talk in mid-April? Oh, wonderful. Excellent. For those of you who didn't come, the purpose of these talks is to familiarize you to get some of the current science of microbiology that's being done with new tools and new ideas out to the public. So the previous, oops, I'm out of sequence, pardon me. Before we get into what the previous talk was about a little bit, I'd like to thank some of our sponsors. We had generous support from lots of people. The College of Arts and Sciences here at the University of Oregon, the ASUO, the Associated Students of the University of Oregon, the Department of Biology, the Institute of Molecular Biology, the Institute of Ecology and Evolution, all provided support, as well as the Oregon Humanities Center's Endowment for Public Outreach in the Arts, Sciences, and Humanities, as well as GREBS, that's us, the Graduate Evolutionary Biology and Ecology students. As budding academics, we as graduate students feel very strongly that it's important to get science out to the public. So we've sacrificed our spare time and our free weekends to put this together and bring this to you. So without all of these people, this wouldn't be possible. So let's have a hand and thank all of these wonderful people that have provided sponsorship for us. Wonderful. So, microbes in the modern world. So we put this together to try to bring some of the cutting-edge science that's happening right here at the University of Oregon out to the public and make it kind of understandable and accessible so that we have an idea as a culture, as a people, of what are microbes, what do they do, where do they live. The previous talk, Brendan Bohannon, on April 17th, presented A Scientist in Whoville, New Perspectives in the Invisible World of Microbes. And he gave us a very global picture of the way we're thinking about microbes today. He discussed how the extent of microbial diversity has been recently, just in the last couple of decades, found to be mind-bogglingly, staggeringly huge. He talked about how microbes are everywhere. You find them anywhere you look, you find microbes. And he talked about how we can change that microbial diversity using examples from his own research in the Amazon. If you missed that talk, it's online. It's free. You can stream it. Just go to our website. If you don't have the URL, it's on the posters, or you can get it from any of us wearing one of these t-shirts, or any of the grad students you see. So that's available. And now, today, Dr. Jessica Green will be expanding on some of those themes, particularly that you find microbes everywhere and that we can change the diversity. We'll talk more, clearly Jessica will talk about that in a few minutes. And then the last talk in our series, in 3 weeks, Dr. Karen Gilliman will be presenting the molecular dialogues with the microbes inside of us, talking about how we interact with the microbes in our guts. So we talk about microbes smoothly from global scales all the way to the little tiny piece of our cells inside of us. Microbes are incredibly important in all of these situations. So, today's talk will be by Dr. Jessica Green. Dr. Green has her bachelor's in civil and environmental engineering from UCLA, and then a master's also in civil and environmental engineering from UC Berkeley. And then she stayed at Berkeley to do a PhD in nuclear engineering before sort of transitioning into the microbial world. She postdoced all over the place, including Sydney, before finally joining our faculty in 2007, where she is now the director of the Biology of the Built Environment Center, some of the work of which she'll be discussing today, as well as running a lab that does all kinds of other really interesting work examining the causes and consequences of microbial biodiversity. She's also a 2010 TED Fellow, as well as being currently a senior TED Fellow. So, everybody, Dr. Jessica Green. change technology over.
Here. Is that gonna? Okay.
Aha. How's that?
That's great. And then I want to make sure. Hi. I want to make sure that everybody can hear me really well. Does that sound good? Okay. I was going to wear what Rue had on, and does this sound a little more clear? Yeah. Although I heard you great, and I really appreciated that introduction. So first I wanted to say I'm very, very grateful to be here, and I think that the image that was used for this series of talks is extraordinary, and I'm honored to be here and also honored to be wearing this awesome t-shirt. What you're looking at now are images of microbes. As Brendan described at the last seminar, these are the most abundant and diverse organisms on Earth. And so by now, most of you probably think of microbes in a warm way, but most people, when they think of microbes, they think of the bad ones, the ones that can kill you, like MRSA or anthrax or HIV. But most microbes are good for us. Like all ecosystems, we need microbes to survive. Microbes allow us to harbor energy from our food. They help boost our immune system. They fight away bad germs or pathogens. And recent evidence suggests that they can even influence our moods, our levels of anxiety and depression. And so if you're having a fantastic day or you're feeling really well right now, like I am, you can thank your microbes. Where do we get our microbes? We get them from lots of places, and we're right now trying to figure out what are the most important sources. But we know that we get our microbes from our mothers. We get them from our food. We get them from our friends and family. And we also get them from our primary habitat, which are buildings. All of us are going to spend 90% of our lives indoors, and while we're indoors, we breathe in and come into contact with many different types of microbes. And because of this, how important microbes are for our health and well-being, and how intimate we are with them in our primary habitat, something that I've been wanting to learn about is, how do we manage microbes in buildings? And to explain how we manage microbes in buildings, I'm going to make an analogy to a garden, a microbial garden, and a microbial garden that's indoors. Modern buildings loosely follow four microbial gardening techniques. These are the opinions of myself, and I'm not subjecting any of my colleagues to these opinions. The first rule that we follow in our buildings is to quarantine the building. Modern buildings are commonly hermetically sealed, and the model is to keep the outdoors out and to keep microbes out. So operable windows have been replaced by elaborate mechanical ventilation systems. And the second rule of thumb is to have doors that allow invasive microbes to come in and be planted into the garden. And by invasive microbes here, what I mean are microbes that are commonly found on human skin and in our mouths. An individual human, a colleague of mine, Jordan Peccia, just published a study a couple of weeks ago predicting that one human in 1 hour sheds is responsible for 37 million bacteria alone being contributed to the air. And humans, that's more bacteria than the number of people that currently live in the state of California. And we know that microbes they contribute microbes to indoor air by directly shedding them from their body, but the majority of microbes that humans contribute to the indoor air are from kicking up microbes that are left behind by people that were in the building beforehand. The third rule for our modern indoor microbial garden is to keep a static environment, a homogeneous environment. Most modern buildings are kept in a very narrow band of temperature and relative humidity. And so the model is to keep our garden without allowing for natural variation in temperature, relative humidity, and even daylight throughout the day or during the seasons. And the reason we keep buildings in this thermal comfort zone is so that if you want to, during the winter when it's snowing outside, you can hang out in your underwear inside. And I don't know how many of you feel like it's a little bit cooler in this room than it necessarily needs to be, but this is an example of most buildings are kept around 69 to 71 degrees. And the last principle is to regularly kill everything in the garden. The way we do this inside buildings is by very constantly using antimicrobial cleaning products and sterilization techniques. And when you think about this, we would never, in a garden that we were growing outside, if there was one weed, kill everything. And I've heard a colleague coin the term microbial genocide as the approach that we're using to try to keep microbes at bay in all of the environments that we inhabit. And from an ecological perspective, we know that as soon as you try to disinfect a surface or remove all of the organisms from one space, what it does is it makes room for potentially highly invasive and fast-growing organisms to grow in that space. And so that very well may be what we're doing, the way that we're constantly sterilizing surfaces inside of buildings. What are the consequences of these 4 gardening rules? Oh, did something just change up there? So, hey, we're back. All right, so the consequences of living in these hermetically sealed, environmentally static, hyper-clean environments, we really don't know what the consequences are. But when you think about it, humans, we've been around for hundreds of thousands of years, and it's only been about 60 years since we lived in these highly modernized spaces. And this came about when the air conditioner was built. It fundamentally changed the way that we design buildings. And I believe that this is causing, basically we're growing a monoculture of microbes indoors. And it's possible that our bodies have not evolved to function in this type of environment. I'm not suggesting that we should go back to a time where we have unsanitary conditions, but it's my sense, and I've discussed this with many colleagues, and people are beginning to write about this, that through our modern way of living, we may have gone so full spectrum in the opposite direction that it's hurting our health. And emerging evidence is suggesting that our fear and ignorance of microbes is contributing to things like antibiotic resistance and asthma and allergies, and the increase of that we face in modern society. What does this all mean? What I envision is that today we have landscape architects. These are people that design outdoor landscapes, and we have groundskeepers that maintain these landscapes and think about the performance of these landscapes over time. And I believe that it's not going to be long until we have a new generation of architect, a new generation of a landscape architect that's an interior landscape architect. And that we're going to have a new flavor of a building manager that's an interior groundskeeper that will be working throughout the life cycle of a building to try to foster the types of microbes that we want indoors. I'm not alone in thinking about this new field of microbiology and building science. I'm working with a team of wonderful people at the University of Oregon and a newly funded center called the Biology and Built Environment Center. And Brendan Bohannon, who spoke before me, works with me at the center. Charlie Brown in the architecture department, and many of the students that are here today have been intimately involved in our system, in our center. And what we're interested in is links between the design and operation and the performance of buildings, thinking at it in the context of energy and also microbiology. I noticed something happened with the mic. Am I still sounding okay? Great, good. So watch this movie to think about how we are currently viewing buildings as ecosystems. That was one of our first collaborative efforts at the Biology and Built Environment Center, where we worked with Ex Vivo, an animation company, to try to help people think about buildings in a more dynamic and biological way. And what you could see in that video, and really inspires me, is thinking about all of the causes and consequences of microbial diversity indoors, which is a fundamental question to ecology. In ecology, one of the main quests is to understand how and why communities assemble. And this is true for not just microbial communities, but also for plants and animals. And what I want you to do is imagine a very simple situation where on one side of the slide you can see a source pool of organisms where the different shapes and colors are meant to represent different species. And on the other side of the slide, you're looking at local communities, which could be islands in an archipelago, or they could be fish guts, or they could be rooms in a building. And what we want to know is how and why might you get different groups of organisms that come from a source pool and are found in a local community. And in ecology, there are two primary mechanisms. There are more than just two, but the two main ones that people think about a lot are the role of dispersal and also the role of environmental selection. So you could imagine that a dispersal process of getting microbes from some source pool to local communities, it could be random, or there could be some type of dispersal limitation like a mountain or an air filter, or any kind of barrier that would keep organisms getting from the source pool to the local communities. And another force or mechanism that people think about is environmental selection. So there may be certain environmental conditions or habitat types that play a role and really allow certain species to grow and certain other species to not grow. So we're trying to understand the relative importance of these two factors in the built environment. And in the built environment, one way that we're thinking about dispersal is through two primary mechanisms. One of them is dispersal from the outdoor air into the building, either through windows or through some kind of mechanical ventilation system, which may or may not be filtered. And the second dispersal mechanism for microbes into buildings is from people that are coming in and out of the buildings. And you can also imagine in buildings there are different environmental conditions that really may foster the growth of some kinds of microbes and not others. So plastics versus wood versus different temperature and relative humidity might play a large role in who lives there and who doesn't. And the first question we sought to answer in the center was really, how does the built environment microbiome, which is this assemblage of microbes in a building, how does that happen in hospitals? And one of the reasons we were interested in hospitals is because it's a classic question that was addressed and thought about over 150 years ago Florence Nightingale, who's my hero because she invented the Venn diagram, she also was a nurse, and she recognized this link between building design and microbiology and human health a very long time ago. And she said the first rule of nursing is to keep the air as pure as within as pure as the air without. And when you think about that concept, this is a very different model. She was advocating for opening windows in hospitals. It's a very different experience than you have it when you go to a hospital today. So most hospitals that you go to have inoperable windows. It's very rare when you go into a space in a hospital where you're able to open the window. So we've moved away from this model that Florence Nightingale introduced a long time ago. And something that we were interested in is, well, does it matter? Is there any difference at all in healthcare facilities if you use mechanical ventilation or if you allow air to directly enter a building through windows? And one of the reasons that hospitals are important to understand is because the rate of hospital-acquired infections is growing every year. There are 3 times the number of deaths in the U.S. attributed to hospital-acquired infections than highway fatalities. Going to the hospital today presents a significant risk that you're going to get sick or even that you're going to die. And so there's very little known about microbial community composition in hospitals, and this is something we were interested in exploring. And fortunate for us, Providence Health and Services is very progressive. We worked in a suburb of Portland at Providence Milwaukee Hospital, who very much wanted to know what would happen in a hospital if you had natural ventilation versus mechanical ventilation. And the reason for this is because a lot of hospitals are interested in going green. A, because they believe that it's going to save them money. As an example, mechanical ventilation is very expensive. And B, because there's an idea that people are finding that sustainable design, even though people don't understand why, tends to be correlated with health and well-being. Hospitals are a fantastic environment to carry out field work because they are, in many senses, homogeneous. If you want to have a room that is replicated over and over and over again with the exact same dimensions, same design, same furniture, you can find that in a hospital. And this is looking at, from the outside, several of the rooms that we sampled at this hospital. We had 3 different treatments where we were sampling indoor air in the hospitals, in this hospital, with rooms that had been mechanically ventilated, rooms where the mechanical ventilation was turned off in a wing of the building and those rooms were window ventilated. So this is a hospital that has been retrofit where you're not meant to open the windows, and they allowed us to do that for this particular study. And we also sampled outdoor air at this hospital. And we had 4 to 5 replicates per treatment, and we looked at both microbial and environmental data. For mechanical ventilation, this is what you're looking at, business as usual, where air comes in from the outside and it passes through the HVAC system, it's filtered, and it goes directly to a room, versus natural ventilation, where we blocked the air coming into the rooms that normally would have come through the mechanical ventilation systems and opened the windows. And the way that we looked at microbial diversity was we used liquid impingers, where you suck air into water, and then when you get back to the lab, you take that water and pour it over a filter, and you can extract the microbial cells from that filter and then extract the DNA from the filter. And for this study, we used PCR amplification and looked at a gene that's phylogenetically informative that tells you something about the evolutionary relatedness among those microbes. And we focused on bacteria. We could have looked at fungi or we could have looked at viruses, but bacteria are the most common human pathogens in hospitals, and that's why we decided to focus on bacteria. This is an image of one of the hospital rooms. The first time we did this study, we worked in rooms that had patients in them because we wanted to have a realistic view of what was going on. And what we didn't realize is that it's a highly dynamic and variable situation when you have patients in a room. You can't ask the nurses, may I please keep the door shut for 2 hours while I'm taking my sample? And even though all of the patients we were working with signed something to let us say that we want to be a part of your study, I could tell that it was making them feel unwell to have people running in and out and keeping these samplers running. They were quite loud. And so what we ultimately did is decided to simulate patients being in a room and to try to standardize things where we had 2 members of the Bio-BE Center with the door shut sat in the room throughout the duration of the study, and we randomized the people in the different rooms so that we weren't measuring the effect of having Jessica in a room, for example. So you can see these 2 liquid impingers over the hospital bed, and you can see this wooden, what would I call it? This big piece of wood that's over where the air comes out through the ceiling, where we were actually forcing the air to go, the mechanically ventilated air to go out of the window. Is everything okay? And it went down to the sidewalk for those poor people walking down the street. And in each room, we looked at several different environmental measurements. We looked at temperature, relative and absolute humidity, and we also measured airflow rate and the air changes per hour. So those little pieces of tape that you can see in the window, the architects that we worked with had anemometers in front of the window that measure air speed, but they also took video footage, which I thought was extraordinary, and then they would go back over time, and they had a process of looking at whether the tape was flapping this way or this way, so they could really tell when air was moving in and out of the window. These are some of our data. What you can see first is that mechanical ventilation does a really good job keeping pollen outdoors. So what you're looking at here on the x-axis are the 3 different treatments: mechanical ventilation, window ventilation, and the outdoors. And on the y-axis, you're looking at the proportion of chloroplast sequences in our data, which is a metric of how much pollen there is. And you can see that the pollen count is much, much lower in the mechanically ventilated room. But what we also found was that the mechanically ventilated rooms had a much lower amount of bacterial diversity. So I want you to look at the x-axis of this graph now. The green dots are our outdoor air samples. The red dots are samples that have been from air from rooms that have been window ventilated, and the blue dots are rooms that have mechanical ventilation. And this measure of diversity, it's a phylogenetic measure of diversity, but what you can see is outdoor air has the most different types of microbes, and indoor air had the least amount of bacterial diversity. And the second thing that we did was we took all of our sequences and we said, what fraction of our sequences are closely related to potential pathogens? And we saw that the sequences from the mechanically ventilated room had the highest proportion of sequences that are closely related to known pathogens, and that the outdoor air samples had the lowest number of sequences that were closely related to known pathogens. And then we further explored this data by asking, what was the indoor air simply a subset of the outdoor air? So we took this data and we put it into an ordination diagram, which is a statistical map. Every dots on this sample, when they're closer together, dots on this graph, they're more similar in their community composition, and samples or dots that are farther apart have less similar types of bacteria. And what you'll see is that the green dots, which are the outdoor air samples, and the blue dots, which are the mechanically ventilated samples, we're not just seeing that the mechanically ventilated rooms are a subset of what we have from outdoors. And the open window rooms are somewhere lying in between outdoor and mechanically ventilated. And when we looked more closely at the data, what we saw was that a lot of the indoor air samples had sequences that were very similar to what you would find on human skin. And it makes sense. And we also found that the outdoor air samples had bacteria that were very closely related to what you would commonly find outdoors on plant leaves and in dirt. And the results of this study have really motivated us to understand more about the human microbial cloud and what we're bringing to the built environment. And these are not images of the human microbial cloud, but they're images that when I think of us shedding microbes, this is what I see. These images were taken by Gary Settles at Penn State University, and it's a type of photography called Schlieren. I'm probably butchering that. Schlieren photography. It's German for streaks, and what it does is it allows you to visualize regions of gas that have different densities. And what I'm hoping is that at some point we'll have the technology where we can really see the density and the spatial spectrum of our microbial cloud and how they interact with one another. This is another one of these really interesting photographs, and the reason I put it up here is we've been batting around ideas in the Biology and Built Environment Center of how we would quantify the human microbial cloud. And as a joke, we've been talking about, it's sort of a joke, sort of not. I don't know how many of you are familiar with the cone of shame that dogs wear, and we've been talking about making a cone of shame for Brendan Bohannon's arm, where we could try to measure the different types of microbes that flux off the human body on surfaces that are at greater and greater distances away from the human body. Now, I talked about the role of dispersal from humans in the outdoors and the environment as the basis of understanding ecological communities. And we did see that there's evidence for environmental selection or environmental filtering in this building. So we saw that most of the variation in the data was explained by ventilation source, but we also found that relative humidity and temperature did play a role in describing the variation of what types of microbes were in the building. And you can see on these plots, what you're looking at here on the x-axis is the relative humidity of different rooms, and on the y-axis is the air temperature. And what you're looking at are isoclines that show the relative abundance of particular microbial groups. And what you'll see is that the relative abundance of the sequences that we found on our air samples were a function of these different environmental conditions. And what that has motivated us to think about is very much in the flavor of architecture, where there's a thermal comfort zone that's designed for, that has this range of temperature and relative humidity and airspeed that we're meant to be comfortable with as humans. Is there a similar microbial comfort zone that's where microbes seem to really foster the growth of particular types of microbes inside? We may continue work in hospitals because it's important, both just for thinking about hospital-acquired infections from a community ecology perspective, and also because hospitals expend an extraordinary amount of energy. They spend 2.5 times as much energy, or use that much energy per square foot than offices do, and they're the second most energy-intensive industry in the United States. And ultimately, what we would like to do is think about the ecosystem of hospitals the way that you might think about a national park, where we can control the growth of some organisms and inhibit the growth of others. And this concept we've loosely coined as architectural yogurt, wondering if there's the possibility of actually instilling probiotics in buildings that foster the growth of the kinds of healthy microbes that we want. And I've given this talk convey this idea elsewhere, and I've been contacted by 3 companies that do this in Europe, where they have a spray that they spray into buildings, and they won't tell me what the formula is, but they say that they have records that people have calling less sick days per year, and they have all these metrics of worker performance. And so maybe this idea isn't very crazy. Before, I want to move on now to talking about our most recent work, which was done in this building, in the Lillis Business Complex. This is the view of the Lillis Business Complex that we see outside of the building. This is the view that a microbe might see. This is taken by an infrared camera, and the different colors show the temperature gradients from hot to cool on the image. This is what the Lillis Business Complex looks like to an architect. And these tables right here were a major field station for us, where we loaded filters into our button samples to sample air throughout the building. So you'll see a closer view of these small filters that we put in these buttons, and the O-ring that we put on the buttons. And then we would cap the buttons for use later throughout the building, but we put these vacuum pumps and filtration devices all throughout the Lillis Business Complex. So this is from the back of the room where we're planning our field day that we were sampling 24 hours a day here in Lillis, and what I thought was extraordinary was that our architectural students like to take the night shift. They like to be up from 10 at night till 6 in the morning, and the biology students tended to like the day shift. This is an example of these filtration devices that we put throughout Lillis. We needed to make sure that the vacuum pumps weren't loud and disturbing the students and the faculty in the building. And so people at the BioBE Center worked very hard to make them quiet. And when we set them out, it was the week after the janitorial staff here had been trained on what to do if they saw a suspicious-looking box around. And so even though I had gone and talked to all the janitorial staff, as soon as we got things going, the calls to the campus police started, and I said, I told you what we were going to do, and they said, I know, but we were told that if we didn't call that we would get in trouble. So we set these boxes to sample air in the corridors. We had them running in classrooms. This is Steve Kimball. You can see through the slit in the classroom. He's a member of the BioBE Center. We sort of babysat our samplers while they were running, which meant that we got to listen to extraordinary lectures I had never thought I would sit in on topics that I've never heard of. That was fantastic. We also had samplers outside to collect outdoor air. And I'm going to show you some very preliminary data. The main question we wanted to ask at the Lillis Business Complex, this is a great experimental system because it's a Silver LEED certified building. And what that means is that we could ask, what is the microbiome of this building like when it's operating the way that it is meant to be? And then what happens if we don't allow it to operate in the most sustainable way, and we explore the air microbiome that way? And so what we did is, on the first floor, we ran it the way the building was designed to be run, where we let air in out directly from the outdoors through louvers during a process at night that's called the night flush. And then on the second floor of this building, we did not allow the night flush to happen, and we basically ran the air conditioner throughout the evening. And what you're looking at is you're going to see an indicator scale on this graph where pink represents bacteria that are commonly associated with human skin, and blue represents bacteria that are commonly found in dirt and on plant leaves. And what you'll see is that on the first floor of this building, you see that the microbes look very similar to what you would find in dirt. It's a very similar story to the hospital story. And on the second floor, you see microbes that look more human-like. And we're in the process of analyzing this data right now. The sequence data is still coming back. We also did not just looking at the two different floors of the building under these two different treatments, but we wanted to think of the whole building as a meta-community where every room is an island. And we sampled dust in all 300 rooms of the building to get a better picture of the historical map of microbes in this building. So I'm going to show you some pictures of people vacuuming Lillis Business Complex to get microbes. This is Steve Kimball. We sampled microbes everywhere in places that Tim didn't necessarily want to be. And the protocol for sampling microbes was to vacuum for 2 minutes at a surface that was, optimally, it would be above head height. The third thing we did in the Lillis Business Complex was we wanted to study one classroom in great detail. And we wanted to see how much spatial variation you might see in an individual classroom. And so we made a microbiology modified Whittaker plot for any of you plant biologists that are used to censusing plant diversity. And we put these Whittaker plots all throughout one classroom. And I just want to show you, we took surface swabs from all throughout the classroom. And this is an example of where I took a surface swab. You can see here with this infrared image in blue was where I swabbed the surface of the table to get the sample. I'm going to close with telling you a little bit about a project that is very related to what we're doing at the Biology and Built Environment Center. Something that we don't know very much at all is really the relationship between the microbes that we're contributing to the building, whether the building is really contributing microbes to us, and how much the interaction among organisms influences that. And so, as an example of an extreme environment that we've considered working in or been asked to work in, NASA contacted our group and wanted to know if we wanted to get involved in sampling the International Space Station as one of the most extreme built environments to try to study. And this would be a really interesting environment because it's a closed environment where humans are interacting, and I think that we could get a good idea of this relationship between ourselves and the buildings. And another project that we're looking at is related to organismal interactions, and this is my way of making forging a bridge between this talk and what Karen Gilman is going to be talking about next week, or in 2 weeks. So can you turn the volume up?
From the time we're born, we're forming a unique self, one that is ours alone, distinguishing us from the masses. We call this our identity. We create it by our choices, by our dreams, and by our character. We wear certain clothes, live in certain places, we choose certain professions, and we forge relationships with certain other individuals. Some of us may even join movements that reflect who we are or what we aspire to be. Roller derby is one such movement, shaping the identities of thousands of women. We might think this new identity comes from a sense of camaraderie, or from the subculture they're building, or simply from their shared excitement over the sport.
But could it be a result of something we're not seeing? When we move down to a smaller dimension, we see a very different reality. We have 1 trillion human cells that make us who we are, but each one of us carries 10 times more microbial cells. These microbes are not passive riders. They do all sorts of amazing things. Many of them even keep us alive. Think about it for a moment. 90% of what you think of as you is actually microbes. It's widely accepted in the scientific community that our microbial cells may be more personally identifying than our human cells. Yet, you're exchanging microbes all the time with everyone you come into contact with. If your microbes define your identity more than the cells that make you human, could the transformation in identity these women are experiencing be due to the microbes they're sharing? Are we really distinct individuals? Or are we just part of a vast, overlapping cloud of microbes?
Is there such a thing as individuality? Or are we actually all one?
If you want to see a great talk about organismal interactions and a wonderful model system, please come to Karen Gilliman's talk on May 29th, and thanks so much for having me. Yeah. And I'm happy to take any questions. Yes? Oh, I just saw this. I just saw this in the New York Times Magazine. The whole raw milk movement in California, it's really big. So I got excited about the topic. But I think what you're asking is, are we doing any work on kind of trying to identify the role of the built environment versus food versus interactions with people? And we're not directly doing that at the Biology and Built Environment Center, but we don't know the answer to that question. We don't know what is the most important for the human microbiome, and I think it's important to figure that out. Yeah, so that's a really good question. Yes?
Is all outdoor air pursuant to your study the same? Like some places outdoor air seems to be very different than other places.
Yes. So I spend a lot of time here, and I just went to New York City last week, and I realized at that time that we may be sampling the Pacific Northwest phenomenon, where this may be an environment where it makes a lot more sense, for example, to open windows, but if you're living in New York or Bangkok or any megacity, you could have very different results. And so something that we have been talking about is really trying to replicate these kinds of studies in different regions of the world. And there's two different approaches that you could have for that. One is, let's kind of get things moving here locally where it's a lot easier for us to do the work and hope that people begin replicating these studies around the world, or we can try to do that ourselves. But that's a really good idea. Yeah. Yes?
I don't know because I don't study microbiology, but it seems to me that pollens and dust and particulates that would be found in polluted air or in aggressively organic air would be bigger than most microbes.
So pollen is bigger than most microbes, and that's why it gets so readily filtered out. And what you're asking about is really the particle, whether or not the different sizes of airborne particles, what role that plays in the types of microbes that you have. And that question is exactly what Anne Womack, who is here tonight, is researching. So she just bought a cascade impactor, which will allow her in outdoor environments to filter microbes of different sizes and look at how the composition of what's there varies depending on size. And one recent study came out that suggests it matters a lot. And the reason that's interesting is because different particle sizes disperse in different ways. And so if it's true that you can really see signature compositions at different particle sizes, then this can tell us a lot about how and why you might expect to have different microbes in different places. Yeah. Okay. Oh, yeah. Yes. Well, it's funny that you asked me that because Ford called me recently, and they are designing, the hyper green car. And what a lot of people want to do now is they want to go, companies, they want to go green and they want to go healthy. So they are asking questions on, because the car is a built environment, exactly that. What's the best way to design the air conditioning system in a car? What kind of material should I have on my steering wheel? How we brainstormed about thinking about the successional life of an automobile. So all of the components come from different parts of the world, and all of those pieces are getting colonized by different microbes before it even gets assembled in the factory, before it even goes out and is bought by somebody. So we don't know the answers to those questions, but Ford is thinking about it. Yeah.
And what do you know about those plastics products that are antimicrobial and how they would affect your studies necessarily?
I don't know. We haven't done any direct study with antimicrobial products, but it's definitely something that we're interested in doing, and it's an obvious question. People are really beginning to think deeply about what we're growing on them and if it's what we want.
I think a lot of people would fear keeping windows open in like a hospital just because they fear that they're inviting new pathogens in.
Mm-hmm.
But have there been any studies done where, like, if people have seen if they've actually reached like a breaking point where opening a window isn't going to let in any more pathogens than there already are? Like the ones that have a significant affinity to humans are already there, so that's not really.
So I'll be very honest right now. The reasons why we operate buildings the way we do now are a mystery to me. I've done a lot of thorough research on this, and I have yet to find really strong, compelling evidence or a motivation for using a mechanical ventilation system versus it depending on the situation. So there are some environments, I mean, if you're operating on somebody or if somebody has an extremely compromised immune system, there are examples of when you might want to do that. But from what I've been told by people like Robin Gunther, who designs hospitals, and people working in infection control, is that there's one published study that showed, I think it was tuberculosis, that actually migrated out, from one hospital room, out of it and into another one, and that was one motivation for keeping windows sealed, was actually protecting people in the hospital from airborne stuff coming out of the hospital and entering back into the rooms. I don't know if that answers your question, but I think because it's been only recent that scientists are using molecular tools to look at the whole diversity of microbes versus one individual airborne pathogen, we don't have the answers to a lot of those questions. And I'll say one more thing. Depending on what kind of infection that you're concerned about, airborne infections, of course, are driving a lot of the code on the way that we design buildings. But from my personal experience, most of the infection control people that I speak with think that the real risk is through human-to-human contact and human-to-surface contact of transferring pathogens, for the majority of pathogens.
So I've become pretty wary of human stewardship on any level.
Human stewardship?
Yeah, so stewarding a specific environment to cater to a specific type of bacteria or plant or anything else. I've heard through the years a number of different stories on a more plant-based scale about there was on the East Coast a bug was introduced to take out multiflora rose, which is invasive, and it seems like you're kind of hinting at wanting to get rid of the bad microbes. But it seems like in creating an environment that caters to the good microbes, can there be a point of, like if I ate yogurt every single day, can't that get to a point where your body just kicks out the rest of them and maybe there's not so much of a benefit on that level?
Should I try to rephrase your question?
It didn't make sense.
No, I think I know what you're getting at. So I think what you're getting at is that it's important that, or why don't I say what I would advocate for? I would advocate for having a better understanding of how the types and diversity of microbes influence whether or not bad or good ones can colonize there. I mean, at this point, we don't even really know how to classify good microbes. I think that, I mean, Karen may touch on this with her talk. We certainly know some of the microbes that are bad and that can kill us, but this definition of what a good microbe is something that we talk about all of the time, and that's something that we're trying to figure out how to quantify. But by just focusing on the bad ones and by killing everything to get rid of the bad ones, work on the human microbiome has suggested that that's not necessarily a good model. But I'm not advocating for one plan or another. I'm just advocating that we study it. And yeah.
I think your point about diversity is really good, but the one word that I guess I would think about was immune system.
Mm-hmm.
That connection between diversity and the immune system. Is that possible?
Yeah, so there are students here. Keaton, where's Keaton? Well, Karen's lab group is studying exactly that. So they're looking at the interrelationship between the immune response of a model organism and microbial diversity. So, and there is a motivation for doing that, to think that they're coupled. I don't know if that answered your question. Yeah. Yeah. Yeah.
Do you foresee a time where you develop studies of microbes to the point you could begin to synthesize them to attack one another?
Okay, so this is going to be my last question, and I want to get it right. So can you say that again?
There's some countries that are doing very green agriculture.
Yep.
They're actually developing microbes that attack some insects.
Oh.
So I'm asking, will there be a time where studying microbes, you begin to synthesize them or develop them?
Oh, biosynthesis of microbes. There probably will be. I don't know if that's something that my lab will be doing, but I think that this idea of synthetic biology, and particularly in buildings, there's a colleague of mine and friend, Rachel Armstrong, who's thinking about that a lot, and how can we use synthetic biology.
Good or bad idea?
Oh, I had to have a loaded question on my last one. I don't know. I don't know. Yeah. Yeah, I think I don't know. So I'm going to end my session on an I don't know. Thanks.